This is the 2026 International Cannabis Business Conference coming to you live from Berlin, Berlin. Here to lead our next panel on the topic which I will reveal to you right now. The topic of which we will discuss, which is known as making products that are GMP compliant, right? Because who doesn't love paperwork? To lead our conference right now is the CEO and president of Cannabis Consulting Limited. Let's hear it for Constant Ma. Constant Ma. And everybody, come on over, you guys. Come on up. Get out the way. Quit making a fire hazard. Come on up, Constant. Thank you very much. Appreciate it. You got a mic.
How's everybody doing today? So thank you for joining us. We're going to be talking about making our products, or making your products, GMP compliant, and the technicalities of it, and how our expert group of panelists here, who has their own manufacturing of decontamination equipment and products, they're here to talk about some of the things that they're able to do to help mitigate some of the risks that cultivators might face, or exporters might face as well with their products. So myself, my name's Constant. I'm the president, CEO of Cannabis Consulting, and we specialize in quality compliance, focused mainly in Canada, but we are doing international GMP framework as well as JCP as well. So I'll do the round of introductions. We have Matthew here, and you can...
Hi. I'm Matthew van den Born, CEO and founder of Flower Power North Macedonia. We are a producer of medicinal cannabis. We're GACP and currently working on our clean room to become GMP compliant. So I'm one of the growers, actually producers of flower, and next to me is George, who is in equipment to help us to reach the GMP standards for flower. Please, George.
Yeah, so I'm George. I'm not going to stand up. Everybody, we had a long day, all right? I'm George Terry. I'm the Executive Vice President of RadSource Technology. We are the #1 world leader in on-site decontamination using X-ray. And so it's an alternative to gamma and E-beam.
Good morning. My name is Rui Soares. I am a founder and CEO of Paralab Green. We are a company based in Portugal and Spain, but working worldwide. We started 34 years ago, mainly supplying pharma industry at Iberian level, but since 2018, with the advent of medical cannabis in Europe, in Portugal, we start offering a complete product range for post-harvest extraction and purification. And being pharma our previous life, so GMP compliance is one of our most strong subjects.
Joseph Edwards, biochemical engineer. I represent Cannabis Technologies as the Chief Science Officer and Vice President. We're the new player on the board. We're a US-based company specializing in organic decontamination and GMP consulting.
I'm Ed Wells. I'm the Vice President of Sales for Canatrol. We actually are in decontamination. We make post-harvest technology to control the drying, curing, and storage process for cultivators, giving them the level of control they haven't been able to achieve historically from any other method to have predictable outcomes.
Yeah, and all of this is super, super important because with GMP, we have to make sure that our products is consistent at all times, and we also have to make sure that it's safe for the patients that are using it. So, and with cannabis cultivation, it's very difficult because it's a non-homogeneous product. It's living, so it's different batch to batch, cultivator to cultivar to cultivar is different as well. So it poses a unique challenge in cultivation and manufacturing, and I would like to talk to Matthew because he's currently going through that journey right now from already GACP, now going to GMP. There's some interesting insights that you've learned throughout your journey, maybe some do's and don'ts, perhaps. What were some of your challenges that you faced, maybe managing microbial loads? I guess my question to you is, during your process of, or during your journey of becoming GMP, was there any process changes or mitigation steps that you guys implemented because of increased microbial loads, for instance, or any way to manage the quality? Was there anything that you've done within your operations to do this type of improvement?
Well, I must say that we are in a luxurious position that almost all the batches we've produced so far are within the contamination range. So we were quite lucky because we were very strict from beginning on with cleanness, with hygiene, with protocols and everything. The only challenge we have is actually in the indica strains with botrytis. These huge buds are very hard for us to keep within the limits. Especially because we like to do everything hand trimmed. So we do upside down drying. So that's a process where a lot of people come in the same room. So we try to get the HVAC adjusted to that amount of people, like 30 people in the same room, a lot of flowers there. We have batches of 800 flowers a day. So we try to make those small batches with other people there. But I think we have some people that I spoke to Rui yesterday actually about this. He said, well, I have some tips for you there to get the contamination rate as low as possible in that first process. And then we have like 5 more steps where things can happen. But please help me out here. What do you suggest to keep the bioburden as low as possible in that process?
In trimming?
Yeah, well, trimming or deleafing, the first stuff. Yeah.
No, yes. One important point is in terms even on the production area, and I have to say this is not our area is post-production, post-harvest, but taking care, especially before the harvest, to deleaf is very important. You have gains in terms of potency of flowers if in the last 2 days you totally deleaf, but yes, in coming to the trimming, coming to the drying, coming to the trimming, depending if you have hang drying or rack drying, so wet trimming or dry trimming, this can be in different sessions, drying or trimming. But coming into the GMP area without leaves is important. And it comes now also the question if hand trimming is better because you get a better trimmed flower, a flower looking better, but at the same time you also are getting more human contact with flower, and so the risks of contaminations increase. This is one of the points to consider. I don't know if you want to say something more. I like then to address this, but I think Matthew has much more to say.
Yeah, well, so that's the first part of the trimming process. After that, our deleafing process is still in the GACP area. Then we go through a pass box to the GMP area. There we have our first 4 drying rooms. We dry upside down. In that process, there are also some steps we can take, like UV light or whatever, but as we work now, we're still on the safe side. Then we go to the second trimming process, hand trim, make the buds small, and then we go to the next 4 drying rooms where we do drying and curing on the racks. And especially there is, you try to do it as slow as possible to get all the terpenes right, do the curing. Those are the processes where a lot can happen before you pack. And also this gentleman over here, if one of these steps might get you into trouble, I'm looking there at the end, somebody is nodding.
Yeah.
Help me out, what can we do to keep this as sterile as possible?
Well, for us, it really comes down to, at the heart of GMP, you're identifying risk, and then you've got to do your best to mitigate that risk so you have safe, predictable outcomes in your product at the end. So for drying and curing, you, chances are, have that bioburden in your flower coming from the cultivation side, coming out of the grow. So that count can continue to grow, and your bioburden is going to increase until you get it to shelf stability. the name of the game, and from our perspective, is to provide an environment where that flower is going to become shelf stable as soon as possible, because that is your window of risk in your dry cure process. When your flower is at a water activity that's going to support microbial growth, that bioburden is going to continue to grow. So, there's a lot of different methods. Rui talked about maybe mitigating how much you're handling it and trimming it before, because that's more touch points, more risk of contamination. There's a lot of different things you can do to, again, reduce non-value adding touch points. It could be contamination points as well. But truly just getting that flower to shelf stability and holding it there by controlling your vapor pressure in that space is really key to controlling that bioburden level and stop it from growing. That being said, there's still going to be a lot of chances in batches where that bioburden exceeds your accepted threshold, and then you do need to move into another process after that, which is where a couple of these other gentlemen kind of come into.
Well, and to support what Ed is saying, I think one of the chief touch points and pain points that we see as we go through our consulting processes with our clients is that the explosive microbial population growth or potential for that growth as we come through that harvest scenario and achieving that active water content. And one of the things that we often advise our clients is that to look at it as a parasitic host relationship. While we may have initially low bioburdens on a product while it is live and thriving, once we cut that product down, biological life, like all life on this planet, attempts to replicate when it sees the end coming near. So having an environment that is both stable and secure through that drying and curation process as we achieve that ideal active water content is kind of that first chief main step to mitigation for what we see as post-harvest bioburden as it goes into final packaging. And one of the things that we can really do to push that forward is controlling that scenario to an nth degree. And as Ed was mentioning, it really does come down to vapor pressure.
Yeah, absolutely. And to shorten that time, that risk window, there are things you can do. You mentioned hanging the plants, and then you break them down more, and then you're drying them on trays. So the more you break them down at time of harvest to have the smallest amount of biomass connected to the flower still, the faster the flower itself is going to come to equilibrium. So we've tracked this in our own trials in the U.S., and what we see is it's going to take the longest if you're hanging full plants. Kind of a no-brainer. Historically, people were really hung up on hanging full plants because the whole low and slow, you thought you had better control. That is true when you don't have control over your environment. So your plants get to a point where they're dry enough, and then they get to a point where, oh no, they're way too dry. If you can slow down that process, it was like a crutch to help you with not having proper control over the environment. So on the flip side of that, when you do have proper control, breaking it down more and reducing the amount of biomass that's still connected to your buds before you go into that drying process is going to speed it up. So what we see is if you did a full wet buck and you went into the beginning of your drying process drying on trays, you're going to be at equilibrium and shelf stable in like 4 days. If you're hanging full plants, depending on the size of them, it could be 6 or 7 days. You're doing a modification somewhere in the middle where you're breaking down to like 18 to 24 inch, we call them spears or branch lengths. That's going to be somewhere in the middle. But then there's other decisions that are going to impact that, because we can say, okay, we'd really like to break them down fully wet and speed that process up by only drying buds on trays. But what is your labor force able to achieve the day of harvest? Are they able to break that down all at one time to get it loaded? Or they bring it in and then, like you said, a couple days into the process, continuing to break it down. So while there's things you can do to mitigate, understand there's operational challenges for that as well.
Yep. Yeah, I also want to point out that you asked me a flower mistake that I made. I come from a growing background. I used to be a grower in Holland for like 20 years. So the first thing I wanted to do is grow as fast as possible, get these growing rooms up and running, show my investors a proof of concept, look what we can do, look how we can grow. And I forgot to put all this effort in the clean room, in the layout of the clean room, all these consultants that I needed to prepare my team, find a proper QA, QC, QP, get all the documentation ready. So then we had this product. At the moment I have like 1.5 tons on stock. And we don't have a proper line to get this all done and get it out on the market. So my tip to whoever grower is here or thinking of starting a facility, focus on your clean room first. Make sure that your product, whenever you got it ready, is directly ready for the market, because building a clean room is one, getting it validated is two. And then you have these gentlemen here that might come up with a solution somewhere halfway your process that you have to implement in your clean room, like a sterilization process or a bioburden reduction, as they call it. Then your whole GMP process has to be reviewed and revised and see how you get it in. So yeah, that's one of my flaws. I focus too much on growing.
Yeah, and from our perspective, I've been in hundreds of grows across multiple countries. Aseptic practice is key. You know, getting that in place early, understanding what contaminates your flower and when it contaminates the flower, whether it's during the grow or it's during the cultivation or it's during the processing. And for Pete's sake, don't leave your flower out, you know, after after it needs to be out, bag it, you know, because you're going to stop the infiltration of microbes into your flower at that point. And then don't reach a dirty hand in there to grab some out and use tongs, you know, set up good aseptic practices for the handling of your flower after you've grown it and cured it.
And I'll continue that. I think those aseptic practices, while handling is an absolute major key, it also comes down to how we look at facility design, air handling through the facility, sanitization of that air, and the process movement of the flower through it. If we're looking at even proper PPE usage through employees, if we're not having an aseptic practice through air handling, air exchange, sanitization, and isolation of those kind of molecular room environments, and looking at everything from a microenvironment standpoint. The same way that we look at cultivation through flowering, we need to begin looking at that too through drying, through curing, through even hallway passages. Everything takes an effect. If we don't have clean air, if we don't have clean practices, clean facilities, we have the risk of, as Ed was saying, until we hit that active water content, a high degree of cross-contamination potential. And this is one of the big touch points and pain points I think that many of our clients see.
Yes, it sounds like proper planning is really the first step in beginning your GMP journey, because as you can hear from the experiences from the group of experts, that if you don't have proper planning at the very beginning, you can potentially be introducing a lot more time into your project that you weren't planning for, and also a lot more cost. And as we all know right now, capital is such a hard thing to come by in our industry, which actually brings me to my next question. To these experts here. As we know, capital is very challenging. Is there, for a company looking to get their GMP certification, is there any specific processes in which cultivators would have like the most bang for their buck in terms of implementing some sort of remediation that sees like way better benefits at the very end with the amount of capital that they invested? I'll ask you. Yeah.
So going through the, you know, your vendor does your IQ and your OQ to make you ready for PQ. You know, some people with self-certify, that takes a long time, and you got to work with the vendor to make sure that your installation, your operational, you know, checkpoints all match what they anticipate as the outcome of your product, so it's always consistent. So, you know, capital to get invested in is basically just trying to get a good consultant. We use a consultant out of Canada called RQC. They work with us. They go through our quality department. Some people do their own self-certification, which is basically what it is. I mean, the GMP is self-certification against the European Pharmacopoeia standard, and you know, once you self-certify and you say that I'm consistent as a vendor, I can always produce the same result, whether it's my software or it's my process or the way I approach the outcome of the flower or the application itself. So if you know, spend the money on a good consultant. If you don't know it too well, at least for the IQ/OQ, or have the vendor work with the vendor in order to make sure that the vendor knows what they're doing and they're able to self-certify.
And again, George, I think you're exactly right. Risk assessment here is key, and it really doesn't matter where that capital investment comes from on that post-harvest process line, as long as it's coming in the correct fashion. If we're not looking at risk assessment and how that implements into your unique operation, your unique operational methodology, your unique flow, while GMP does give us a fairly linear line through process, there is a large degree of variance in application of those principles. And as we apply those principles, looking at what each individual technology offers you, what each individual benefit might be comparative to your operational needs, I think becomes paramount. And again, to harken back to George, that proper consulting there is absolutely key.
Yeah,
I
well,
would like to.
oh, sorry, Rui.
Matthew, please.
Well, the best tip I ever got, and I really hope that there are people here in the audience now building a facility and trying to build a clean room as fast as possible. The best tip I ever got was cut it into chunks, because I wanted to do everything. I wanted to do edibles, I wanted to do extraction, different types of packing, dry flower, whatever. So my consultant told me, listen, Matthew, just go for dry flower in a package form that every pharmacy will accept, like 250 grams or 400 grams, and that's the process. You first validate, then you get your GMP, and then you do add-ons. Then you go for extraction, whatever. But if you want to do all these disciplines together, there's a bigger chance that you will fail. So just go for your first simple product, dry flower, packed in a certain bag, and then do an add-on. And we did that, and that make it so much easier to do it together with the team. So that's really a tip from me. So just go for a simple dry flower and then do add-ons.
Well, what I would like to say in terms of where to invest. My recommendation, I've doing this for 8 years, and I've seen lots of projects, several that thrived, many too many that went down. Why? What are the difference? I think the main difference is if who invested right or not. And to invest right, what I realize in the industry is if you invest for quantity and not for quality, you have no future. So independent of companies that want, like, I will be producing 10 tons on first, second year, and prepare all the flowering rooms, all the drying rooms, all fantastic till a certain point facility. But then when you start looking, their HVAC system on flowering rooms is maybe not the best and doesn't have proper moisture control, and so you end up always continuously have high contaminations. Your drying rooms, in spite of having Canatrol systems from ED, have ship splits or under-calculated, projected HVAC systems, no curing, simple packaging systems, not under vacuum or inert atmosphere. So all of this leads to bad products, and bad products will not sell or will sell for a very, very low price. So my recommendation is, whatever the budget you have, first look for what is the best practices, the best technologies, and based on your budget, rethink your project in terms of the capacity that you may have the best technologies and to the quality that that budget will allow. But you will have much better chances of strive, of being economically okay, so selling for the price and something that pays for the structure. Quantity in this industry, it has no future. It's quality that has future, because quality will bring you not what have been discussed here, a product without contaminations. Actually, hopefully, and I speak about me because decontamination, bioburden reduction is my main business, but the objective, and especially we have been here talking about GSP and GMP, and there's been a lot about contamination flower, at least contamination. It comes even from JCP. What the guideline says is that remediation, so bioburden reduction, should not be the basics, fundamental process. Like we always know that we will have to do a bioburden reduction at the end. This is going not even against GMP, this is going against GACP guidelines. We should be setting up our facility, our process, to always considering how to reduce any sources of contamination, hopefully avoiding decontamination or bioburden reduction of our material. This is critical, this is one of the aspects, but then it's also, yes, if you have the correct lighting, the correct control of temperature, humidity, and air flows, you will also, the right nutrients. I see people saving money on new nutrients, and this is a huge mistake if you want the right quality of flowers. So this is where people should be investing to get the products desired quality.
So how do you, like for me as a consultant, the biggest question I always ask when I'm proposing a new project to my clients, it's always that value proposition. Like, why should I spend this X number of dollars now? And what is your justification, or how have you guys kind of navigated those conversations? Like,
Well, so I would say not just for GMP, but just in general. We have a saying about don't set yourself up to have to spend good money after bad. So obviously you're going to have to there's a certain threshold you're going to have to meet in how you set up your facility, how you outfit it, the types of equipment you have to meet GMP. That being said, the biggest bang for your buck in this business, whether it's GACP, whether we're talking about a rec market in the US, a pharmaceutical market here, LPs in Canada, look for ways that you're going to differentiate yourself. It's not going to be on quantity, like Rui said, it's going to be on quality. Look for ways that you can give your consumer a better product, a more consistent product. There are efficiencies, though, a way to do that, but also cut your operating costs. Things that are going to set yourself apart from your competition in an industry that is going to continue to compress, because every market, once it's been open, and the more mature that market gets, the more it's compressed, the more pricing compresses. Setting yourself apart and differentiating yourself is always going to be the best place you can choose to spend your capital.
Absolutely. I think what we need to look at is a clean-in-place approach. Environmental security, operational consistency, these lead to a continuity of product quality. And as we look at that clean-in-place approach, decontamination becomes compliance assurance more than it becomes an absolute necessity for product viability.
Yeah, it doesn't have a bad stigma. The people, I hear all the time, decontamination, you should grow clean. Well, if everybody can grow clean, we'd be eating raw chicken, right? I mean, microbes have their way into things. GMP is one of the processes that allows you as a grower to put processes in place to try to fend off as much as you can microbial infestation or even more, that you have the same product, that is consistent. Imagine if every time you took a Tylenol, it was different. That wouldn't be good.
Yeah.
So.
No, we usually use Coca-Cola as the analogy, but same thing.
I mean, it's a drug, though. One day you take Manjaro and you don't eat for 2 weeks. Come on. You need consistency in the process. That's what GMP is about. And then at the end of the process, you need a fail-safe. And whether it's in the curing or it's in the cultivation or it's in the growing, there are solutions out there that can cover all of them.
Yeah, and it's actually, there's only one goal, right? Patient safety.
Exactly.
yeah, that's exactly right. Perfect. I guess that concludes our all the questions I've had for the group today. I can open it up to the audience if you guys have any.
Let's hear it again for Constant Ma and all of the panelists. Let's hear it louder for Constant Ma and all of the panelists. We have questions. They have answers.
I got a question. Where'd you get those shoes, man?
Skechers, baby. They're very comfortable. That's my first advice for traveling is wear comfortable shoes. I walked 16,000 kilometers yesterday.
Wow.
No, not 1,000. That would be a lot. But 16 kilometers. Okay, you have a question there. Here I come. Those shoes. It's classic.
Sorry, could you lift up the hat again? Sorry.
No, it was over here. It's Michael. We were all confused. Pass the mic like a joint.
Cheers. Hi, Paul speaking. A quick question about, obviously, different processes of, let's say, remediation and decontamination. Obviously, like we said, where the efficacy of this is making sure that you want predictable administration of any medicine, right, or drug. So you want to do that for the standards of the person that's utilizing the medicine, should we frame it? So, but what about the buyers when we're looking at different methods of remediation and decontamination? Cryopasteurization, right? But Paralab, you're representing VIST, for example. There's different methodologies, so where does the compression come in there that actually benefits the efficacy of the consumer, but is also serving the needs of what the buyer wants in terms of the standards or what they prefer in their methodology for decontamination?
Yeah, it was a little hard to hear you
Okay.
up at the top, but I think you're asking about the different methods of decontamination. So during the stages, during your cure, during your grow, you have different methods that are out there in order to try to keep it down. And then at the end, you have different methods for post-cultivation. But the idea is that, no matter what method you use, once that flower is ready to go to market, it's sterile. It's clean, and it's clean enough for consumption of the user, and there's minimal impact to the terpenes, the cannabinoids, the consistency of the feel, the touch, the smell. So there's multiple different methods out there. I think everybody here is GMP compliant. Yeah, everybody on this panel is GMP compliant as far as our methods. And it's just up to the consumer to pick out, have a discussion with us.
Yeah.
And we'll explain.
What's best for your operational profile? What's best for your needs? How do we fit best in your operational flow? Everybody here has, George mentioned, is compliant with 5.1.4, 5.1.8b. How does that fit into your unique process, your unique post-harvest chain? And how do we look at that from a viability standpoint for continued operation, continued product health, and that downstream leads to continual quality for both the consumer, patient, as well as the producer?
Well, I've got one question there, if I'm allowed to. How do the end buyers react to this? Because it's written down in the process, there's full traceability. Buyers can see there has been a process for bioburden reduction. Is there a preference, or does it go just like all the other flour?
Well, it has at this moment, by German regulations, there is this bad perception for irradiation. We have here George. I have to say that irradiation is a very good technique. It's just this German regulation that created this bad perception of irradiation, and that basically, in terms of market, people selling, made life of George very complicated. Saying that, his life would be complicated with the advent of cryopasteurization, because irradiation, it was until now the only technique able to do 6 kilo-log without damaging much the flower, still affecting. Cryopasteurization came as technology that gets there even damaging less. At the same time, does not have this market perception of irradiation. People think that irradiation means radiation and gamma radiation, the product gets radioactive, something like that. It's crazy, doesn't? But there is this perception. And yes, this created new companies to come to the market, and somehow this was a driver to come with new technologies that have fantastic performance with at least 4 reductions of 3 kilo-log kills, 4, 5 log kill, have basically zero impact on the flower. So preserve terpenes, preserve color, preserve morphology of the flower, and have this very good, this very high decontamination. George, I don't agree with sterilization. We do not have sterile flower. That doesn't exist.
So the other half of that is, yeah, there is a stigma out there about irradiation, especially when they irradiate your meat and your eggs and your chicken and your fruits and vegetables out there that keep people safe. But it's a perception issue, because the reason that people use irradiation and the reason that irradiation is the gold standard for sterilization is because it works. it works. And what has to happen is the consumer's got to understand that there's different types of a radiation. For example, you have gamma radiation, you have E-beam, you have X-ray, is what we actually use, and you have UV. I mean, anything that you treat visible light, we're being irradiated right now. Anything on the electromagnetic spectrum is radiation. IR, RF, all that stuff is radiation. And people, we have the American Nurses Cannabis Association that we've talked to as well. We're a sponsor of them. They say, people that are going through radiation treatment have a stigma about radiation, but the bottom line is radiation works. It's been the gold standard for 30 years, and there's other technologies out there that work as well.
All right, thank you. That's perfect. We're right on time. So I appreciate you guys for keeping to the perfect schedule. We got like 8 seconds left. Let's hear it for the panel. Make some noise. We're going to take a picture. We're going to move on to the next one. And I'm sure if you have more questions, they will answer them very in depth